Thank you, Greg, and thanks to everyone for sticking around to the very end. So we're going to finish up with my favorite topic, which is immune therapy for myeloma, and I will try not to go too long. And so we're really fortunate that almost 2 decades of research into how the immune system recognizes cancer is now finally leading to new treatments to try to target that immune system. And so what's been known for a while, not just in myeloma but in cancer in general, is that the cancer cells don't just grow in a vacuum. They interact with other cells within the tumor microenvironment. And actually, the cancer cells learn to subvert this immune system. So our immune cells can actually recognize our cancer cells, but they can't eliminate it. As the immune cells learn to become the cancer cells become resistant to those cells and can actually turn the immune system against itself. And so these decades of research have now actually taught us what types of tools we can use to try to overcome this resistance to the immune system, and now actually have led to a number of approved therapies that actually target not the cancer cell itself, but the surrounding immune cells to try to wake up that immune system to fight the cancer. And so this has opened up a whole new sort of way to treat cancer and is now being applied in myeloma. And so I'm not going to be able to talk about all these approaches today, but we can discuss some of them in the question and answer. But I'm going to focus on a few. One is the use of what's called cellular therapies, and I'll explain what that is, particularly one called CAR T cell therapies. I'll talk about another type of antibody called a bispecific antibody or a bispecific therapeutic engager, a BiTE, and how that works. And I'll talk about a novel type of antibody called an antibody-drug conjugate, or ADC, and explain what that is. I'm not going to focus too much on the standard monoclonal antibodies you've already heard a lot about from Pete's talk, or about vaccines or checkpoint inhibitors, but you can ask in the question-answer session. I'm happy to discuss them. Okay, so if we're going to try to target use immune therapy to target myeloma, what target should we use? So you've heard about some of the targets of elotuzumab and daratumumab, these markers on the cell surface. But the one that really is showing a lot of promise for a lot of the immune therapies is called BCMA, B-cell maturation antigen. And this is a protein, a receptor that sits on the surface of the myeloma cell. It's also expressed on our normal plasma cells, a subset of our B cells and other immune cell and dendritic cells, but really not expressed on other normal parts of the body. So this makes it a very promising target that it will really try to focus primarily on the myeloma cell. And it's expressed almost universally in myeloma patients, though the intensity of expression can sort of vary from patient to patient. But you can stain for this on the myeloma cells, or you can test by something called flow cytometry and show that these are 5 different myeloma patients and their myeloma cells all express BCMA. And BCMA also can be cleaved and released into the circulation, and so you can measure soluble BCMA in patients' blood, similar to the way we measure free light chains. And there's not a clinically approved assay yet, but there's one in development, and hopefully this will be another way we can measure if patients express high levels of BCMA. And importantly, BCMA is important in the survival of the myeloma cell. So this is very nice data from Ken Anderson's lab at Dana-Farber. If you knock down BCMA in myeloma, the myeloma cells start to die. If you force high levels of BCMA in a myeloma tumor, it'll grow faster. And so again, this really makes this a rational target for immune therapy. And so there are a number of different approaches for targeting BCMA. I'm going to go through some of those. The first one I'm going to talk about are the so-called antibody-drug conjugates. And so most standard antibodies like daratumumab or elotuzumab are so-called naked antibodies. The antibody binds to the surface of the myeloma cell and marks that cell so that an immune cell, like an effector cell, natural killer cell, can come in and try to kill the tumor. And this drug can do that as well. But what's different about the antibody-drug conjugates is that attached to the backside of the antibody is a very potent chemotherapy-type drug. In this case, this particular drug called GSK2857916, it's a chemo drug called MMAF. And once the antibody-drug conjugate binds on the surface to the BCMA, it's internalized into the cell where that chemotherapy drug is released. It's sort of like a targeted warhead. And then that chemo is concentrated inside the myeloma cell and spares the rest of the body the toxicity if you just gave the chemo through the vein. And so it's a way of really targeted delivery. And that leads the myeloma cell to die. And then it can also block signaling through BCMA. So this antibody can actually work through multiple different mechanisms to try to kill myeloma. And so based on preclinical data that demonstrated activity in myeloma cells and in mice, there was a first-in-human phase 1 study with this antibody-drug conjugate that we participated in. And this looked at 30 different patients in the first portion 70% 5 or more prior lines, so very heavily pretreated patients, 90% double refractory to IMiDs and proteasome inhibitors. And the drug is given as a 60-minute infusion every 3 weeks. And we started, as most phase 1 studies do, at very low doses and then slowly escalated the dose up. And the first portion was really to look for safety. And one of the things we were concerned about is what's called corneal toxicity. So this chemotherapy drug, MMAF, that's on the back end of the antibody, even if very small amounts get in the circulation, it's known from prior studies using this agent that it can deposit in the cornea and cause corneal irritation. And so patients all got some steroid eye drops to try to prevent that toxicity at the, with each dose of treatment. And this is the data from the phase 1 portion. And so the patients who got the lowest doses are at the top, and you can see there were very few responses in the very beginning at low doses. But as we started to get to higher doses, we started to see fairly remarkable responses, including very good partial responses, stringent complete responses, in 6 out of 9 patients treated at these higher doses. And this is a single agent without any steroids or other drugs mixed in. And so this was very promising. And so this led to a larger study that was presented at this last year's ASH meeting, where we enrolled another 35 patients, I should say an expansion of that prior study, treated at the higher dose, and with a response rate of 60%, including 51% who got a very good partial response or better. And so this was, again, fairly promising, better than we might have expected in a phase 1 first-in-human study with this. And this is demonstrating the percentage decrease in the patient's M protein, or free light chain. You can see a number of these patients, 100% reduction in their M protein going forward. And so the other thing that was interesting was that a lot of these responses were durable. And so this is the duration of patients were on treatment. And if there's a little triangle here, it means the patient's still on study. And you can see getting out more than 8 months, 10 months, a lot of patients still on the drug at this time. And so I think promising initial responses with this antibody-drug conjugate against BCMA. Now, what's the trade-off? So in terms of toxicity, the most common one was low platelets, thrombocytopenia. And this was seen in a little more than half the patients. It's a known expected side effect of the drug. It's reversible. If you hold the drug, it comes back up. And unfortunately, despite the steroid eye drops, the corneal toxicity was still a problem. It was seen in almost 2/3 of patients had some type of either blurry vision or dry eye or light irritation of the light. And most of this was mild and didn't require patients to come off study, but it did require the dose to be held, and in many cases, the dose to be decreased. And some of my patients, instead of getting the drug every 3 weeks, will get a dose every 9 weeks or 12 weeks because their eyes will get flare up and then they'll come back down. And so in general, this is certainly a difficult toxicity. It was fairly manageable in these patients, and based on these data, there's actually a larger randomized study now that's about to open that's looking for registration for this particular drug in patients who are heavily pretreated, who have already become resistant to a proteasome inhibitor, an IMiD, and daratumumab. And so stay tuned, but we're hoping this is going to be another drug, and it's the first drug targeting BCMA as an antibody-drug conjugate showing activity. Now, this is not the only antibody-drug conjugate. There are certainly other ones that are out there in trials. This one also targets BCMA. This one hits another target on myeloma cells called CD48. And interestingly, these use different toxins than the MMAF. And so these toxins don't cause the eye toxicity. They may cause neurotoxicity or neuropathy. And so we have to see you have to pick your poison to some degree. But the hope is that we'll have a lot of these different drugs that are going forward that may become new options for us and relatively easy to give. Okay, so what's the other option that we're using for immune therapy? The one that's generating, I think, the most excitement is something called CAR T cell therapy. So CAR stands for Chimeric Antigen Receptor. And the concept here basically is that you take the recognition domain of an antibody, which is useful to bind to anything on the top of a cell that you want it to, and you create a new receptor that actually can signal and turn on a T cell, so that this is supposed to be the membrane of the T cell, your immune fighting cell. The outside looks like an antibody, so it can bind to the tumor. The inside signals and activates that T cell. And you can use a gene transfer technique to express this CAR on the patient's T cells. We're able to do this outside the body. I'll show that in a moment. And then you add certain co-stimulation domains that can make those T cells grow and live for long periods of time. And therefore, you've generated a whole new set of cells in the body that can recognize the cancer. And so this is sort of a cartoon about how this works from one of my colleagues. The way we actually get the CAR into the T cells at Penn is using a viral vector called a lentivirus, where we've taken out all the guts so that the virus can't replicate, but it's a very good virus that can get into T cells. Once it's there, it delivers the genetic material that encodes the CAR. And now that T cell, instead of recognizing whatever it was supposed to recognize before, now can recognize the surface on the tumor cell. CD19 is expressed on B cells, and so this is the first CAR construct that was used in some leukemias and lymphomas. And then it comes in and attacks the cell, and you get a dead tumor cell. And so the way this works is that patients undergo a leukapheresis. This is similar to when you collect stem cells. You basically sit on this machine for a few hours, blood goes out, we suck off all the T cells, the blood goes back in. Then there's a manufacturing process that takes anywhere from 2 to 4 weeks, depending on the study, where those T cells are transduced to express the CAR. They're grown up and activated to large numbers and frozen down for the patient. During that time frame, many patients continue to get additional chemo or whatever they were on to hold their myeloma in check. Then when the cells are ready, they're usually given a transient dose of chemotherapy called lymphodepleting chemo. That's designed basically to clear out their own endogenous white cells temporarily to make room for the CARs to go in, and then you infuse the CAR cells like a blood transfusion, and they start to expand and grow within the patient and hopefully attack the cancer. And so that's the concept behind this. And for a long time, this was really sounded like science fiction, but actually with advances in the pheresis and the manufacturing, this actually is working. And really the place where it's worked the most and actually is now approved is in certain B-cell cancers, chronic lymphocytic leukemia, acute lymphoblastic leukemia, and non-Hodgkin's lymphoma, where we're seeing responses in about half the patients with CLL and non-Hodgkin's and up to 80% in patients, particularly with pediatric ALL. And this is Emily Whitehead, who's literally become the poster child for this type of treatment. She was one of the first patients treated at the Children's Hospital of Philadelphia with this approach, had refractory ALL, had no real treatment options, and got this, and now is over 5 years later, still in remission. And this led to the FDA approval of these approaches just in the fall this past year. And we have patients still out 5 years or more without it coming back. So this works the best so far in these malignancies, and the question is, will it work in myeloma? Now, there is a trade-off, as there is with everything. There's no free lunch, and so there are toxicities with CAR T cells as well. You can get very rapid killing of all the tumor cells at the same time. That's something called tumor lysis syndrome, if the CARs work too well. Usually we can manage that, however. B-cell aplasia means that you're knocking out some of the good B cells, and the patients are more risk of infection, although, again, we have ways to deal with that. The two common ones, though, that we are dealing with, one is called cytokine release syndrome, or CRS. And this basically happens when the T cells all get activated when they see the tumor, and they all start to proliferate and divide and multiply, but they also release factors into the blood called cytokines. And these cytokines are normally produced when there's an infection, but you can get really high levels of them, and it basically can mimic the worst flu of your life. And so some patients just get high fevers, flu-like symptoms, and can be managed supportively, but other patients can get more severe symptoms, low blood pressure, difficulty breathing, even end up in an ICU. And so you have to know this when you're going into a CAR trial, that these risks are possible. Fortunately, we have sort of an antidote to this CRS called tocilizumab. It's an antibody that blocks one of these cytokines called IL-6, and this has been very effective at treating CRS. The other side effect that we see is called neurotoxicity, and again, this can range from just a little bit of mild headache or confusion for a day or two to more severe patients becoming obtunded, getting seizures, and in some of the CD19 trials, there have been rare brain swelling and even death. And so this has great promise, but also toxicity as well. We are learning now sort of how to predict for some of these side effects, and so they're becoming less frequent in the later trials. We're treating patients earlier with tocilizumab, but something just to keep in mind when you think about these trials and this type of approach. Okay, so we know this works in B-cell cancers. Can it work in plasma cell cancers like myeloma? So the first group that actually developed the BCMA CAR and tested it in mice and then in humans was the group from the National Cancer Institute, James Korkendorfer and colleagues. And so they presented and then published this first-in-human experience. They treated 12 patients with heavily refractory myeloma. They gave them, initially, as I mentioned, some lymphodepleting chemotherapy, so 3 days of chemo to, again, clear out some of their own immune cells to allow the CARs then to go in and expand. and they gave CARs at escalating doses. And they didn't see much at the lower doses, but at these higher doses, 3 out of 6 patients actually had a significant response. And this was associated with rapid expansion of the CAR cells. You can see within 5 days after infusion, they could measure these CARs at high levels in the blood, and they could last for a couple of months and even longer. These patients did develop cytokine release syndrome, and some developed delirium, so some of the same side effects we saw with the CD19 CARs we're seeing here, but this really showed proof of principle that this could work, and some of these remissions were fairly durable. And so at the University of Pennsylvania, we also had been working on developing a CAR in collaboration with Novartis, and so this was our first in-human study with also a BCMA-targeted CAR, structurally a little different from the NCI CAR. We were looking at 3 different cohorts of patients. The first just got the CAR cells alone without chemotherapy at a high dose. We wanted to sort of see what the effect and safety would be just with the CARs, and then we added some chemotherapy, cyclophosphamide with a lower dose of CAR cells, 10-fold lower, and then the Cytoxan with a full dose of CARs. And again, this was a pilot study really looking at safety and feasibility and efficacy, but in addition, we wanted to follow the CAR cells. You can actually see them here on flow cytometry and measure how they expanded and how long they lasted and whether this correlated with response. And so at ASH this year, I presented updated data on our first 24 patients that we treated. These were, again, heavily pretreated patients, 7 prior lines of therapy. They had, almost all of them had seen the usual agents for multiple myeloma. We had a number who were quad and penta refractory, as we heard about in the last talk. Almost all of them were high risk by cytogenetics. 70% had a 17p deletion or mutation, and a lot of myeloma in the bone marrow. And what we saw was that, as in the first trial by NCI, we could get responses with this CAR T-cell approach, even in these heavily refractory patients. And surprisingly, even in the first cohort where we just gave the cells alone without any chemotherapy, which we weren't really expecting. So the patients in blue are those who had an objective response, a partial response, or better. And we had 4 patients in this first cohort, including this one patient who's sort of our poster child. He's now about 27 months, so over 2 years, still in stringent complete remission after a single dose of CAR cells. And he had 11 prior treatments, 17p deletion, and a TP53 mutation. So really incredible. And so we then looked in the second cohort with Cytoxan and a lower dose of CARs, and I think the results were more disappointing when we lowered the dose. And so in the third cohort, which is ongoing, we're seeing, again, responses in 6 out of 10. This 7th patient has actually responded, so it's 7 out of 11 now, again, in these heavily pretreated patients. So showing proof of principle that a CAR against BCMA can work. Unfortunately, what we're seeing is that the majority of patients are not like this first patient. We have some patients still with ongoing responses, but many of the patients get responses that last 3 to 5 months, and then the disease is starting to come back in this setting. And our big push now, I think, is to try to understand why some people are in ongoing complete remission and some are not. And what we really think is that it may have to do, to some degree, with the quality of the T cells we collect in the beginning. And some patients who have been just so beat up with so much chemotherapy may not have T cells that can make the best CAR cells, and perhaps treating those patients earlier before they've had so many lines of treatment may help. That's our hypothesis, but we need to test that. As with the other trial, we still did see severe cytokine release syndrome in 33% and severe neurotoxicity in 13%, fortunately reversible, but again demonstrating that the significant toxicity of these cells as well. The side effects, however, tend to occur within the first few weeks. And so once you get past that initial period of the cells expanding and then coming down, we haven't really seen late or long-term side effects. Now, there are several other studies of CAR T cells that are ongoing, and a couple were presented this past year, and this is the one at ASH this year that really generated the most excitement. This is a study from a company called Bluebird, and the data were presented at ASH this year by, again, James Korkendorfer from the NCI, who developed the original CAR construct at the NCI. And so this is also a BCMA-directed CAR T cell. They reported data on 21 patients, again, very heavily pretreated, 7 lines of therapy. Not quite as many double refractory patients or high-risk patients as our study, but certainly patients who had been through almost everything. And I think what's exciting about this trial, they didn't see a lot when they used the lowest dose of cells, but once they got over 150 million cells, 17 out of 18 responded, 94%, including over half the patients with a complete remission. And all of these stars are when they did MRD testing, and at least on their MRD assay by flow, a lot of these patients were MRD negative. Again, almost remarkable for people who are this heavily pretreated. And so they also are starting to see some patients progress. 4 of them had progressed, but a number of them had still ongoing remissions. This is weeks on study, getting out beyond a year, 52, 60, 68 weeks. And so we're all urgently sort of awaiting the next update of this, which will be shown at ASCO this year in June, to see if these patients are still in remission going longer. But I think, again, showing more and more evidence of efficacy. And the last trial I'll mention was this interesting study presented at the ASCO meeting last June. This is a Chinese company called Legend Biotech. And they also have developed a CAR against BCMA. They didn't give a lot of details about how it binds or what the CAR was made of, but they described data on 19 patients who were valuable for response. These patients were definitely not as heavily treated as the American trials, and they didn't describe what they had previously. But again, 100% response rate, 74% stringent complete remission, and these patients are those who are still in remission more than a year, and they have 5 patients so far still in remission. So I think we're going to see more and more that this is an effective treatment for these heavily treated patients. The question is going to be the durability. You saw the first couple trials where they were not quite as durable as the last couple trials, and we have to wait and see if there's differences between the constructs that explain that, or is it just all differences in the patients that got enrolled. But overall, I think this is exciting data. There are still issues with BCMA CAR T cells. I mentioned the toxicities, which I think we're getting better at, and there's trials now of giving this antidote tocilizumab even much earlier, maybe at the first sign of fever, trying to see if we can prevent that bad cytokine release. There are also new constructs within the CARs, something called suicide genes. Basically, it means that if the T cells start to grow like crazy and the patients get sick, there's a gene in there where if you give a certain drug, it makes the cells die. And it may give you a little bit better ability to control those cells if they start to get out of control. I think another issue we may run into is the myeloma cells, again, becoming smart and starting to downregulate or lose BCMA. And we are seeing this in some of our patients who relapsed. Now, it does come back up later, which makes you think maybe you could retreat with CARs or use one of these other approaches against BCMA. But I think we'll need to figure this out or come up with other targets at the same time. And then logistics. CAR T cells are tough. If anyone's tried to get on a CAR T cell trial, there's always a really long wait list and always very few slots. And so it's going to get better as we get more trials, but that can be one issue. As I said, it takes several weeks to manufacture these cells, and we've had people who looked really good when we took their cells, but then their myeloma took off, and they were never able to get them. And then cost, I think, is going to become a problem. The cost for some of these FDA-approved products for leukemia and lymphoma came out between $400,000 and $500,000 for a single shot. And is that really sustainable if we're going to be using this in large amounts of the population? I think if you're curing patients with one treatment, and they're not going to have to go on 4 years of daratumumab, Revlimid, Kyprolis, Dex, then maybe the cost is going to even out. But if you're just getting 6 months of remission and then they're going to go on that stuff anyway, it's not going to work out. So I think there's a lot we have to figure out here in terms of how this is going to be all paid for. But as I said, the field is definitely moving forward. The last time I looked, I found at least 15 trials on clinicaltrials.gov of BCMA CAR T cells. About half of those were in China, and then the rest were in the US and Europe. And so there are a lot more of these available, and I would suggest if you're interested in a CAR cell and they don't have one available at your center, go on the websites, look for other centers, because you may be able to get into a trial somewhere else. I think there's also interest in combining CAR T cells, and we'll have a study open combining CAR BCMA with CART19. CD19 is not generally expressed in myeloma cells, but it may be on a precursor to the myeloma called a myeloma stem cell. And there's some data to suggest using these together may be better than either one alone. Earlier treatment, I think, is going to be next. As I mentioned, rather than waiting till people have had 7, 8, 9, 10 lines of treatment, maybe doing it earlier when their T cells are a little more robust will get better outcomes. And there's this interest in getting off-the-shelf CAR T cells to use genetic editing to allow you to just take CAR T cells that are sitting on a shelf ready to go and then give them to every patient so you don't have to wait those 4 weeks for manufacturing. And that, I think, is something that's being tested in the laboratory now. And then there's other targets I'm not going to have time to go into, but BCMA is not the only target. There are CAR cells targeting all of these that are going into patients. And so we'll have a lot more data, I think, in the next year or two about if this is really going to take off and where we're going to use it. But I do think it's going to fit somewhere in our armamentarium. And then to finish up, the last approach that's being used to try to target myeloma immunologically are the so-called BiTEs, or bispecific T-cell engagers, also called bispecific antibodies. And this is a really clever idea, I think. So basically, you engineer an antibody where one end of the molecule binds to the tumor cell. You pick a target like CD19 or BCMA, something on the top of the tumor cell. The other end binds to the T cell and activates it. And so you're actually bringing the T cell to the cancer cell in the tumor environment and activating it right there and seeing if it'll attack the cell. And so you don't have to go through this whole take out the T cells, manufacture for 4 weeks, put them back in. And remarkably, this can actually work. There's one of these drugs is already FDA approved called blinatumomab, and it's approved for refractory ALL, acute lymphoblastic leukemia, where it has response rates of 40 to 50%. So not as high as the CARs, but it definitely can work. And so there are a number of companies now developing these for all different types of cancers, including myeloma. None of these have presented any data yet, so I don't have any clinical data to show you. It's all data that's basically in mice, and they're now going into patients. But at least in this one study, for instance, they compared a bispecific antibody against BCMA to a CAR against BCMA in this mouse model, and they got the exact same results in terms of tumor control. And so whenever it lights up, it means the tumor's growing. This is the control mice who didn't get any treatment. The mice that got the CAR or the bispecific, didn't have a lot of cancer here. So there are at least 4 of these targeting BCMA that are now entering trials. There's another one hitting another target called FcRH5 that we have open at Penn, and then several others going forward. And I think, again, this may be another useful approach because this is, again, off the shelf. You don't have to manufacture anything. It's just infused into the patient. Now, sometimes the infusion can be a pain. Blinatumomab has a really short half-life, so the patients have to wear a little pump that continuously infuses it into their PICC line or vein over a 4-week period. So there's some logistical issues to work out. But again, this may be another option going forward. And this is the comparison of all these approaches. So I think the CARs are not off the shelf. The antibody-drug conjugates and BiTEs are, so that's really good for convenience. The antibody-drug conjugates are really easy to give. Any community oncologist can give it in the clinic. It's a 1-hour infusion, whereas the CARs are only at specialty centers, and the BiTEs can be cumbersome. The nice thing about the CARs, though, if it works, is you could get a one-time dose and you're done, whereas the others, you have to repeatedly dose these and stay on them. And then the toxicities can be different as well. The BiTEs have been associated with some CRS and neurotoxicity as well. Although, again, the duration of toxicity is short for CARs and could be ongoing in the others. So I think there's pros and cons to all of these approaches. I don't think one is necessarily going to win and the others won't be used. I think it's likely we're going to be using all of these approaches and really open up the type of immune therapies for myeloma. And so to quote my myeloma expert, John Snow here, immunotherapy is coming. I think, I didn't talk about PD-1, PD-L1 blockade. We can discuss in the question and answer session. May not have a role in myeloma, but BCMA is really the main target. And CAR cells and antibody-drug conjugates really have high response rates. The bispecifics are just entering trials, so we have to wait and see. And a lot of questions remain. I think, which patients should we be using for this? How are we going to manage the toxicities? How are we going to sequence and combine it with our current therapies, which we already have a long laundry list of good treatments that work. But these are good problems to have as we get more options for our patients. And I think I'll stop there. Thanks.